Welcome to the Simons Institute. My name is Prasad Raghavendra. I'm a senior scientist at the Institute. The Institute is a collaborative institute that was set up 10 years ago with a generous grant from the Simons Foundation. It hosts two programs in theoretical computer science and related areas every semester, and each program attracts about 70 researchers from all over the world. And most programs have 3 workshops and a boot camp. One of the programs this semester is the Computational Innovation and Data-Driven Biology. Thanks to the Cornell Foundation for support on this program. And welcome to the second workshop of this program, Computational Challenges in Very Large-Scale Omics. First, I'd like to thank the organizers, Roderick Guigo, Stephen Brenner, Sandrine Dudois, Tal Pupko, and Ron Shamir for arranging an excellent set of speakers for this week. Before I hand over to Roderick to introduce the workshop in a moment, a couple of logistic things. Snacks and coffee breaks are outside, and lunches are on your own. Please enjoy the food and coffee outside the auditorium, as food they're not allowed inside, but water is definitely fine. Masks are strongly recommended, but not required. Lockers are available on the first floor if you'd like to use them to store your luggage or bags during lunchtime. The Wi-Fi is, of course, Cal Visitor CD-ROM. Our videographer is Omid Fur, who will assist with speakers with microphones and computers right there. And finally, a big thanks to Kelani Penland, who's right outside the door, our events coordinator, for all her work on local arrangements for this week. Feel free to ask us or her any questions that you might have during the course of the workshop. But without much ado,
So I'm Stephen Brenner. I'm faculty here at Berkeley, and I'm joined by Ron Shamir, who is from Tel Aviv University. And we are the PIs of the Core at Berkeley Tel Aviv initiative, which has sponsored the month-long program that's here, of which this conference is a component. The Core at Berkeley Tel Aviv initiative in computational biology has many activities. This has been one of the premier ones to bring people together from around the world to discuss computational biology and bioinformatics. We also have major activities in giving grants for groups jointly between Berkeley and Tel Aviv, and there's been a lot of exciting research that's come out of those, as well as workshops and other exchange programs. And so we're really grateful to Roderick and Sandrine especially for bringing together the incredible set of speakers for this week. And I'll hand it over to Ron. Anything else you want to add?
Thank you, Stephen. So we are finally doing it. Thank you, Roderick, for persisting. We tried it in 2020, we tried it again in 2021, and we are finally doing it in 2022. And, you know, over the last few months while planning it, we were crossing our fingers to make it happen. And it's really great to see everyone here. I should mention that this is part of a summer program of the Correct Berkeley Tel Aviv initiative, and for those PIs and students who are staying here for the full summer program, we are also organizing short talks and reading groups outside the conference weeks. And you are all very welcome to participate. Yeah, I think you will receive emails about this. The first conference that we had 2 weeks ago was excellent, and we are looking forward to another excellent conference now. And thanks, Sandrine and Roderick, for leading this.
Thanks. I have a little to add. I'm very happy to see all of you here after all, as Ron said, after all this year. I actually checked today, and I realized that the first message that I got from Ron and Stephen inviting Sandrine and myself was 3 years ago in September 2019. So it has been really a long journey. So I'm actually happy that almost all of you could make it. We had at the end only two issues with two people that couldn't make it because of COVID, and I apologize in their name. This is Jason, who was almost on the way to the airport, and Jasmine Fisher. Anyway, I believe that we still have really a great workshop, and I hope that you really enjoy it. We had, as Ron said, a great first workshop organized by Rodet, and I hope that this will be of equal interest and that you also enjoy the Berkeley campus. I can be here now for 2 weeks. I'm really enjoying the first workshop and the recovery after the first workshop. Let me just remind you that in addition to the talks, there will be a few additional activities. We have today a panel discussion, which will be led by Sandrine, and followed by a dinner for speakers, poster presenters, and long-term participants. Tomorrow, we had a poster and a reception, which actually will be attended by the Chancellor of the University of Berkeley. And on Wednesday, we have a hike. I'm told that's really a beautiful one. So there is still slots available in case you want to join us. It's about 1.5 minutes hike, so that I think it's probably accessible to all of you. And I just want to finish by thanking the people that have made this possible. Of course, all of you for being here. Then Sandrine Dudois, actually, she has been really a great colleague and has a great help. She put a lot of effort into organizing this and a lot of great ideas in terms of participants and activities. Of course, Ron and Stephen for pursuing this over all this time and for organizing this series, and the Simons Institute. I think this is really a great place, and in particular, I'd like to thank Peter and Kelani, because they are really very responsive of all our needs. I do believe that the lunch today is included, although lunch, I'm confused about this, but right. So in general, lunch is on your own, but I think that today, if I am not wrong, lunch is included because we have really a short lunch break today. Okay, thanks a lot. And well, actually, I'm here because I'm going to chair the first session, and the first speaker is Hagen Tilgner here. Which I don't have the title with me because it's there, but I guess that we will see it here, which is probably single cell in the brain, all right, and long read, things like that. Okay, Hagen, I think that we have, how much, 20 minutes or 30 minutes? No, it's... So
I thought it was 30 with questions.
Yes, 30 with questions. So when you have 20 minutes, I'll try to...
Wait with me.
Yeah.
Okay, great. Yeah, first, thank you for having me here today. I'm really honored to be part of this. And I like to start my talks with showing this cover, which was actually made when I was a PhD student in Roderick's lab, 2012. It was for ENCODE, and it showed many things. So the cover actually goes on. There's things here and here and here. But what was most important to me and to my work at the time was this connection basically between the DNA, the RNA, and the timing of splicing with respect to where the polymerase is. So what you see here is all these exons and these rocket-shaped things here. And then the introns in between are mostly already gone. So while two still remain. So we basically said most of introns are removed cold transcription. Yeah, we're based here in Manhattan. This is where the school is. This is where postdocs are housed. And yeah, we're always looking for good people. So one more introduction about splicing. So splicing is basically the gibberish that the process that takes the gibberish that is pre-messenger RNA, decides where exons are, removes the introns in between so that we get a final message. It was night in October. You can all understand that phrase. With just a little bit of alternative splicing, we can get a second exon here and get a second message. Okay, it was 9th in the lonesome October. Not very different from the first message, but now there's a connotation because the second one is coming out of a poem by Edgar Allan Poe. Okay, so that's one way to think about alternative splicing. So the slight modulation of of a message with a connotation of something different. Now, there are other ways to think about it as well. If you consider this phrase here, Monday I have facts, with just a little bit more of alternative splicing, I can turn that into Monday I have alternative facts, phrase made famous by Kelly and Conway. And of course, if you've seen that interview, you know that this means pretty much the opposite of the first. Right? And that is not just happening in this toy example, that actually happens in biology quite a bit. So there's work coming out of Harvard, this is Mark Vidal's lab, where they basically asked how different are two protein isoforms encoded by the same gene. One being here, so this is a dissimilarity, one being totally different, 0 being the same. And here they frame this in protein-protein interactions. So when you're at 1, the two isoforms don't have any similarity in the proteins they interact with. When you're at 0, you have the same. And you see that for about 20%, you get pretty much the opposite. So basically no protein-protein interaction partners in common. Then for another 45 to 50, you get quite some differences, and then you get less and less. And maybe for 16, 17, 18, you get no differences at all. And I would argue that this is probably a lower bound, because this is obviously done in some condition. And the conditions that we could potentially explore are infinite, or almost infinite. And of course, protein-protein interaction is also not the only thing. So I started my lab 6 years ago in New York City, and as part of a neuroscience institute. So we really wanted to work on this beast here. And in reality, that means we worked a lot on this beast here, and now we're actually starting to work on this here. And so the important point is that whenever you measure proteins, RNA expression, splicing, DNA methylation, or many other traits, fundamentally what you're measuring is sort of a linear combination of signals coming from neurons, from ependymal cells, oligodendrocytes, astrocytes, microglia, I forgot, endothelial cells. And of course, these have more subtypes. So what we really wanted to do is to untangle this linear combination for splicing work. And for gene expression, a lot had already been done. Right? So the whole single cell approach, I think most people know that, was already out. And so we were basically sitting on top of this approach and created something called single cell isoform RNA sequencing, SCISOR-seq. And this was led by Ishan Gupta and Paul Collier, but with contributions by many. So look at the author list, there were people all over the world helping. And so fundamentally what we did is we sit on top of this single cell world, where we basically do the single cell short read approach to get this table that basically connects individual cells or barcodes to cell types. And here's a graphic representation of that. What we do then is we do long read sequencing on these full-length cDNAs, locate the barcodes at the end and the UMIs and so on. And according to those barcodes, we will say that this molecule originates from this individual cell. Now, this cell, according to this table, was already placed in a cluster of cells that we already labeled as microglia, according to marker gene expression. Therefore, we can say that this is a microglial read. And likewise, for the second one, we would go to neurons and would find that this read is now a Purkinje cell read. Purkinje cells, so we're in the cerebellum, part of a very important part of the brain, more the ancient part of the brain. And these cells, by the way, are very famous because they have these very big arbors. So the first thing when we started this, this was somewhere in 2017, we asked ourselves, do we actually do well with recognizing these barcodes? So we took these barcodes that we knew to exist from the Illumina sequencing, learned an error profile with PacBio and Nanopore, and this is here the PacBio data, introduced these errors and asked how often do we actually get the right barcode. And it turned out that the specificity was basically 100%. Of course, when you simulate things, you put a lot of assumptions in, right? You simulate what you think is actually happening there. And so to test that, that this was not some sort of imperfect assumptions that we were making, we also synthesized cDNA, put a fixed barcode in it, which is one of these barcodes, one that occurred very often, put it on a sequencer so we know exactly what is the answer is supposed to be, asked the same question and found again 99.99%. So in terms of specificity, we did really well. I don't have the slide here, and sensitivity, we were not doing so great. And yeah, we have some ways now to get it better. And then with these reads, we can now make these plots here where every line is one molecule. All these lines have been grouped because they originate from the same cluster of cells. And here in blue, we have neuronal clusters. So, and these are the Purkinje cells. And then we have astrocytes, microglia, and OPCs. What you can see is that neurons are characterized by the inclusion of the first exon, the second alternative exon, the third, the fourth, the fifth, or the skipping of it, and then the sixth. So there are two isoforms really characterizing the neuron types. And you don't see these two isoforms, for example, microglia, also not in astrocytes. I think one of them you observe once in OPCs. So what that means is that there is cell type specificity in the isoform expression that is not there at the same level in gene expression, because this gene is expressed to some extent in all of these cell types. Okay, so when we had this technology working somewhere in 2019, we started asking questions about that we thought were relevant biological questions. So here we started with brain region specificity. So, and this was led by a student, Anushka Joglika, who is now close to graduating, but again, there were many others contributing to that. And so what she did is she said, well, if we have P of C in hippocampus, and from either pseudobulk or real bulk experiments, we know that there's differences in isosome expression between the two. What kind of cell types actually account for that? And so there could be one cell type, what could be inhibitory neurons in the hippocampus splice one way, and inhibitory neurons in the PFC splice another way. It could be many cell types, so the brain region could override cell type specificity. And it could be no cell type at all, because due to the abundance and possibly gene expression changes, we could get a very differently weighted linear combination of signals in the two brain regions, and therefore get an apparent difference which doesn't exist in any cell type. Okay, so I'm just going to show you some examples here. These are hippocampal excitatory neurons. You see that this exon is highly included, somewhere 90–95%. In hippocampal astrocytes, it's included maybe 50%. In hippocampal OPCs, maybe 80%. And then you go to the same three cell types in the PFC, and it's mostly skipped. There's a bit of intron retention that could potentially give rise to the inclusion of that exon, but it's much less than you see up here. Now, so that means in this case, really the brain region overrides cell type specificity. And we do see that more often than you would expect by chance, but what's important to recognize, it's still a minority of cases. Okay, so it's very few, but more than you would expect by chance. The majority actually follows this model, where you have relatively similar inclusion of the exon in hippocampal astrocytes and PFC astrocytes. Likewise, similar inclusion in the OPCs in the two brain regions. And then you go to excitatory neurons and you go from 81 to 23%. Okay, so that is really the majority of cases where you have one cell type explaining it. And of course, now we could play the same game at finer cell types again. Yeah, in this data set, we did not. Good. So, and also in this paper, we developed something we call SliceOseq for slide isoform sequencing or spatial isoform sequencing. And basically, this was based on the 10x technology again, Visium, but it could in principle be done with any spatially barcoded cDNAs. So here the barcodes are used not to delineate a single cell, but an area in the brain. So every circle here gets one barcode, and the whole thing is more or less like 3,000 separate sequencing experiments, and we basically developed the long read approach for it. And so here's an example, clathrin A. Yellow means very high inclusion of this exon, black means skipping, and you see a lot of inclusion here in the hippocampus, and then skipping in the choroid plexus. And the interesting point here is that it correlates very nicely with leaving one structure and entering another structure. And the second exon is mostly skipped, but there is inclusion only in the hippocampus, not so much in the choroid plexus. So this is an example that actually sort of goes against what I just said, because here we have, so in SNAP25A, again, one exon here, we don't have this precise correlation with brain structure. So we have a lot of skipping in this area. And as we slowly go towards the hippocampus and the cortex and so on, we get higher and higher inclusion, but it's not really at the boundary of one structure to another. And these are mutually exclusive exons. So the other exon does really the opposite. So, and currently we're involved in the Brain Initiative and doing a lot of work there, where we're basically going across brain regions and developmental time points. Yeah, but this is not for today. So as I told you early on, that really we wanted to work on the human brain and then did a lot of work on the mouse brain. And fundamentally, the reason for that was that in the mouse brain, since you have fresh brain, you can isolate single cells. Right? In frozen brain, which is pretty much any human brain you can use for research purposes, you cannot follow that approach because things are frozen and you cannot isolate single cells anymore. Hence, people use single nuclei sequencing, which for gene expression works just fine. For us, it's a big problem because in nuclei, you have a lot of retained introns and you have a lot of RNA species that don't exist in the cytosol. So it took us a long time to get this working. We call this approach now single nuclear isoform RNA sequencing. It was led by Simon Hardwick, Wen Hu, and Anushka Joglekar. But again, many others contributed to it. And so fundamentally, if you are doing single cell long read stuff, if this is your gene, this is the kind of molecule you would like to have. Okay, you have sort of a PCR adapter here, then you have your 10x barcode, your UMI, poly-DT/poly-A sequence, depending on which strand you actually sequenced. Then you have all the exons, and then you have a template switch oligo, which is made to, which is used to make the second strand. And then those two are also used for PCR. So unfortunately, there's a lot of these molecules. They have all the nice barcode structure, but they start in an intron. They give you 2 kb of intronic sequence, and then they give you the template switch oligo. So that is very nice for things like RNA velocity, a technique that some people here may know. For us, it's a big problem because these things don't tell us anything about exon combinations or the inclusion of certain exons. And then there's a third type of molecule, which is probably not very well known because in the Illumina protocol, you heavily bias against it. That means even if they're produced on Illumina, you won't see them. But for us, when we put it on PacBio or Nanopore, then we end up with all these molecules here. And they basically originate when the TSO, instead of making the second strand, starts to make a first strand. Right? Then you end up with a molecule that has TSO on both ends. And so basically we devised technologies to get rid of, or not get rid of, but to bias heavily against these last two ones so that we get mostly the first one. The first is that we basically start doing a linear PCR from this black element because the black element is always there when the barcodes are there. And that means we make a lot of single strand molecules that represent this and this, but not this molecule. So that means in this cartoon, we put it as if this one disappears. That's not quite true. It does not disappear, but it's not amplified, whereas these two are. Then we use exon probes that hybridize two exons to select for molecules that have at least some exonic sequence. Okay, and again, in this cartoonish version, then this molecule disappears. Of course, it doesn't disappear 100%, but we get a much cleaner, much more of these desired molecules, and then those we then put on the long read sequences. And so this was done in a human frontal cortex. Here you see all the cell types that you would expect: astrocytes, microglia, excitatory neurons, some inhibitory neuron types, oligodendrocytes, and so on. And so what we observe is that in terms of barcode efficiency, we get a 2.2-fold increase. And in terms of having reads that actually overlap annotated exons, we get a 3.3-fold increase. So together it makes a 7.5-fold experiment increase. So that means an experiment that would have cost $30,000 now costs $4,000. And so this is what we get then. So we get quite often reads with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and maybe some more exons. You see it's not quite perfect because for some reason we don't make it quite to the first exon, but we make it quite a long way. What you see here is that we have basically some exons that are really highly included in astrocytes and a little bit in oligodendrocytes, not at all in the neuron types. It turns out that one of these exons, I believe it's that one, maybe it's that one, I don't know, is associated to autism spectrum disorder. So we looked a little bit into diseases, but just to be clear, this data all came from healthy brains. So the first thing is that we looked at exons that were highly variable. So that when we look at the inclusion percentage, that is the lowest among all the cell types and the highest, we had some exons that have, a change of at least 75 percentage points. And these are these blue ones here, and they're very short on average. They peak at about 54, so it's not only microexons, although they include a lot of the microexons. And then the less variable types are actually having quite longer distributions with medians or with peaks above 100 bases. Yeah, there's work in this area by the Blanco, Gashrin, and Ponting labs, so you should probably also read that. When we then looked at exons that were associated to different diseases, for the autism ones, they were really very highly variable. So exons associated to autism, much more variable across cell types than the ones not associated to autism. In ALS, there was a hint of that, and in schizophrenia, we did not see it. There's one caveat that these exon lists come from different papers, and so the methods to define them are not exactly identical. So yeah, we're still looking into this some more. But now these are autism-associated exons, and now we have basically this heat map again. We can see that this exon here highly included in the neuron types, not so much in the glia. Then you have the ones that are highly included overall, lowly included overall, and here the ones that are only in the glia. And now in disease, that raises very interesting questions, because when an exon is decreased in inclusion in the bulk, in these exons, it could come from pretty much any cell type, or maybe all cell types together. In these, it would stand to reason that it would come here from the excitatory or inhibitory neurons, in these ones from the glia. So that means there's now, for some exons, it's actually very clear. If there's strong decrease in disease, then for this exon, it must come from inhibitory neurons. But for these ones, it could come from pretty much anything. Okay, so now I want to talk a little bit about the combinatorial patterns of all these elements, exons, TSS, and poly-A sites. So here, if you forget for a moment all the cell types that are being shown here, what you observe is a lot of molecules that have both exons included, and a lot of molecules that have both skipped. And then once in a while, you may find somewhere here some molecules that have just the first, not the second. So that means it's strongly biased towards the both inclusion or both skipping form. And that's what we call coordination. And I will use that word from now on in the talk. And so we basically looked at this, the coordination of exons, TSS, and poly-A sites in specific cell types. Here, just another example. This is MAPT, so microtubule-associated protein tau, very important in pretty much all the neurodegenerative diseases. A lot of skipping of both, inclusion of the first, but skipping of the second, skipping of the first, inclusion of the second, but not inclusion of both. So they're mutually exclusive, which is also a type of coordination. And here in this example, again, you have two, if you look at the pseudo bulk, you have two nicely coordinated exons. However, when you look at them in cell types, you can't really say that they're coordinated anymore, because here this exon is not even alternative. So that means in this case, the coordination of the two exons is explained by constitutive use of at least one, almost both exons in the neurons, and pretty much constitutive skipping in all the glia. And so this is what we try to highlight in this heat map here. So we started with exon pairs that were coordinated, and the pinkish color tells you that in this cell type, we can prove coordination. What I want you to observe is that for these exon pairs here, pretty much in every row, you see some pink color. Maybe the last 10% you don't, but that means in the vast majority of cases, we can actually trace this coordination to the cell types. When we do this for TSS exon pairs or exon poly-A site pairs, we cannot. And this greenish color here means that one of the elements has become constituted. Okay, two more slides. So we basically started thinking recently to really understand in detail the error patterns of PacBio and ONT. And so the question that we asked here is, if you put the same molecule on PacBio and nanopore, would the two agree or not? And so this was done, you can't really put the same molecule, but you can use barcoding techniques and then have basically cDNA copies of the same RNA molecule, put them on both PacBio and nanopore, and see where they disagree. And what we observe is that at TSS, there's 80% agreement, but there's 8% where only the PacBio has a TSS and nanopore is not. And there's about 3% where they have different TSS. Now, the last part here is, if this is the annotation, and you have this kind of molecule or this kind of read coming out of the nanopore with an acceptor that is 3 bases shifted up, Now we can ask on the same molecule on the PacBio, what does the PacBio actually say? And in this case, it confirms it pretty much all the time. It's a pretty rare event here, but those upstream NAG-NAG shifts, we basically believe. If we do this for donors that are shifted by 4 base pairs, it's basically never. So basically 1 in 1,000 where this is actually correct. So, and basically we've exploited all these error patterns now to build an annotation pipeline that we call Isoquant, which in our opinion does really, really well for novel isoforms, meaning that are not in the annotation. For known isoforms, it does also well, but there are also other programs that do well. Yeah, and I guess I'll skip the remaining slides and go for questions. And let me just put up the thank you slide. So thank you for listening. This is the group, the people that do all the magic, former members, collaborators at Weill, Cornell, and the funding sources. Yeah, and here are the conclusions. I won't go through them, but
we have time for questions. So.
Yeah, that's very, very fascinating. I was wondering, to what extent in the current analysis you're conducting is the decoding of the barcodes the bottleneck? I mean, you mentioned the error, patterns at the end comparing ONT and PacBio, because the 10x barcodes was designed for long read, high error rate. So is that a limiting factor? And do you need, related to this, the short read data still to match those, or could you do just de novo long read sequencing? Maybe thinking about your last few slides where you compare technologies.
Yeah. So there are 2 questions. The first one, so on the PacBio, it's not so much the bottleneck anymore, because the error rate on the PacBio now, if you have CCS, the circular consensus rate is really low, so it's really not a problem anymore. On the nanopore, we get much less barcodes or high confidence barcodes, so it is to some extent. And so from what I've seen, you can increase recall quite significantly, and maybe even in a way that you decrease precision only slightly. But in my opinion, let's say if your precision drops below 90%, it's already a very, very bad run, right? Because that would mean 10% of your molecules would have been assigned to wrong cells, at least possibly to wrong cell types. There was one question. What was the other one? No.
But you still need the short read data.
Oh, so we do it with a short read data, usually because this is sort of the minor cost in the experiment. Technically, you wouldn't need it if you have enough high quality reads, right? Because you could do in principle, the long reads, you can chunk them up to a fake Illumina read and then do exactly what you did before. What actually turned out for us to be a major bottleneck, which took us basically a year and a half to solve, is this problem of these molecules that have TSOs on both ends, which basically have no barcodes. Because that turns out to be a huge driver of the cost, and yeah, now we're much better with that. Yeah, I think so. Probably, let's say if PacBio has a major increase in throughput, there will be days then on the PacBio you would do this without Illumina sequencing. On the nanopore, I mean, they already have very high throughput, and there's just a new kit out now for the R10 flow cell. It's possible that we'll reach that soon. Technically, you can do it now, but it's just more expensive, basically.
Hi. You have been showing how you could use isoforms to identify particular cell types. So my question is, what will be the relative contribution of specific isoforms to neural cell type identity versus the expression of different genes? And whether you could use this information for deconvolution approaches and so forth.
I mean, theoretically, you can. Well, right now, it's a bit difficult to answer because usually we don't have the same depth per single cell as the short read approach. And so for gene expression, also the beauty is that if one gene gives you no molecules, that's actually information, right? That's you can use that for clustering. If for splicing, if the gene gives you no molecules, it's basically NA, it's not zero, right? So I'm not yet 100% sure what the answer to that is. I hope we'll get there.
Thanks.
I have a question regarding the last part. You didn't talk about the isoquant. So what specific technicalities did you consider to infer isoforms from the long read data? And did you do that differently for PacBio and nanopore?
Yeah, so so we have a couple slides about this. So there is, what it is, basically it's a graph that represents introns, and basically you have a edge between two introns if they're adjacent. Right, and then it uses a lot of techniques out of graph theory. It comes out of the genome assembly field. That bulge removal, for example, seems to be very important. But also what I mentioned previously, that the splice set, that we do some splice set correction where we say, well, look, we've learned already that this kind of mapping is not to be trusted, and then there are other ones that are not in the previous paper where we basically look very carefully at these alignments and ask whether we should trust them or not.
Okay, thank you.
Thanks. I have a more exotic question, and also forgive me if it's naive. I've been out of this area for some years, but I mean, you didn't mention the different types of spliceosomes. I mean, U1, U2 versus U11. Does that have any? I mean, also there are some genes where you mix the two types of introns. Is that something you can study here, or?
Well, theoretically you can. The problem in a sense is that something like, I think 98.5% of introns are U2, and then 1% is U12, but out of those 1%, 2/3 are still GTAG. Yeah. So fundamentally, your observations will always be dominated by the U2 or the, let's say, the GTAG consensus. Yeah. Right? And so we haven't really dug deep into it. The code actually checks, we only believe reads when every read is consensus, which means GTAG, GCAG, or ATAC, which includes the U12. We haven't really ever made a statement about the U12 ones, because my feeling, and please correct me on that, is that in a sense, U12 is super important for some genes, and then for many it's not. That means unless you are really into this gene, right, it may not be so it doesn't lend itself to make genome-wide statements very easily, right? But it could be, yeah. It's if the mapper does the mapping right, then there's no problem with it.
So the same question for UTR introns. Anything that is special with them, or because you didn't mention them?
So UTR introns, we haven't specifically looked at them. I mean, they would be in the mappings. In the papers we published, we did not look at them. We should actually. I mean, what I would say is that probably in the 3' less because of nonsense-mediated decay, and 5', yeah, there's upstream open reading frames. I know we've actually discussed that, but in the end, nobody really mapped these special 5' UTR things so that we could actually check them against introns. But I would assume that there must be something also that, some Kozak sequences, some start codons that are affected by introns. Now, how widespread, I don't know, but I'm sure many, there must be a lot of work out there, maybe not about brain cell types, I don't know.
Hagen, wonderful talk, and excuse me if this is a naive question, but how do you distinguish tissue specificity and isoform expression versus temporal differences, say due to chronology or chronicity rather? How do you know? Are you looking at a snapshot in time? Do you look across different times?
Yeah, so in this data, we haven't. Now what we're doing in the brain initiative is exactly that, to do developmental time courses where mice are being interrogated at multiple time points. So I hope this data will inform us on exactly that. And so we are playing a lot with what we call these hypervariable exons that really change dramatically. It's actually quite interesting because there's different patterns. Some really go up in time. So you just go into your neurons, and then over time they come up. Some peak early and come down again.
So what you're presenting is kind of a snapshot. They're all controlled at a certain you know,
Age,
you
yeah. So
collect
what
them
I
at an
showed you
age and
today,
time and
what I
so-
showed you today in mice was once at P1, 1 day after birth, the cerebellum, and then P7,
Yeah.
I didn't say that.
Yeah.
So 7 days after birth, that was the 2 brain regions. Now on the brain initiative, it's multiple time points that basically describe the.
So we really don't know.
Well,
yet.
we have some data. I haven't shown anything here, but yeah, we have some
Yeah.
data that actually shows many exons that do these things. Yeah, they give you basically certain groups, but everything I showed you today is a snapshot.
Do you
That's correct.
suspect something like an editing mechanism in cephalopods, or you know that you see in cephalopods? You think there's a lot of variability in this, and
Well, I mean, it's already established by many other people before us that there is developmental regulation and splicing in many people focused on neurons, but also in glia. There are some things known. So yeah.
Yes, another question here, Kathy.
I was interested in your result that the autism genetic risk was enriched in the variable exons or the genes with variable exons. And the reason I was is we've been studying the non-coding risk for different psychiatric diseases and see a lot of cell type specificity as well. My question is, do you think that that is because the risk is in cell types that themselves just in general have more exon skipping or variability in the isoforms? Or does your result hold for each cell type? Like if you look at it cell type by cell type, do you see the trend consistently across cell types, or is it sort of specific to cell type?
It wasn't, I mean, what I was expecting actually when we did this is that the autism exons would all be highly included in the neurons and not in the glia. And there were certainly a lot of these, but there were also some that do the opposite. So yeah.
Yeah, we're seeing the non-coding risk in the glia also.
Yeah, so I think that it doesn't seem like there's sort of one cell type where that is pinpointed by here, everything goes wrong. Yeah, but we haven't done the cases yet. But I would guess that it actually, you know, that there's multiple exons that go wrong in just one cell type or just in glia, and then many that go just in neurons, and then maybe that some that go wrong all over the place. That's my guess. But
cool.
Thank you. It's a great discussion, but we need to move to our next speaker.